okay? So respiration leads to the production of carbon dioxide, which is a waste product. The urea cycle is a very brilliant cycle. Whoever designed it is a genius. Takes two waste products, that is ammonia, and combines it with another waste product called carbon dioxide to form urea. So this CO here actually comes from the carbon dioxide that is coming from respiratory reactions within our body. So we take that carbon dioxide and we combine it with ammonia, which is another waste product, and then we release. Of course, there's the other way that you get rid of carbon dioxide through the lungs. Of course, bicarbonate, you goes into the red blood cells, it is converted to bicarbonate, transported all the way to the lungs. Most of the carbon dioxide that you form within your system is transported in blood as bicarbonate. Bicarbonate is hydrogen carbonate, HCO3 minus. That is called bicarbonate or hydrogen carbonate. A little bit of it is transported, of course, as carbaminohemoglobin, right? Yeah, but most of it is actually transported as bicarbonate. So urea formation now is what we want to understand. Why? We have come all the way. We have known, of course, where ammonia is coming from. We have seen how that ammonia is transported all the way to the liver. Now, once we get into the liver, the objective there is to convert it to a form that is less toxic and is soluble, okay? And that form is urea. We've said from here, once we form it in the liver, we form the urea in the liver, we can diffuse it back into blood, transport it into the kidneys. Then the kidneys will filter it out of our system. The story should be very clear in your mind like that, okay? So urea formation, the first step is usually the activation of ammonia. So we've seen where ammonia comes from. So where does that activation take place? It takes place in the mitochondria. And remember, we are in the liver now, okay? These reactions are taking place in the liver. So ammonia is reacted with carbon dioxide to form carbamyl phosphate. So ammonia comes from the amination of glutamate and glutamine. So there's an enzyme that is called carbamyl phosphate synthase, CPS1. That's the enzyme that converts, of course, ammonia and carbon dioxide to form carbamyl phosphate. And the reaction here normally requires ATP and N-acetyl glutamate as activators. So you can see, of course, the reaction here. Carbon dioxide plus ammonia plus 2 ATPs in the presence of this N-acetyl glutamate will form carbamyl phosphate plus 2 ADP and inorganic phosphate. Okay, so if you want to approach it in a lazy way, then you say, oh, the first step is the formation of carbamyl phosphate from carbon dioxide and ammonia. Okay, for now, you know, there are people who want to get the big picture first, and then they go and sit down and internalize everything. So you tell yourself the first reaction, you form carbamyl phosphate from carbon dioxide and ammonia. Okay, yes, but if you are you know, getting the details all the way, you need to pay attention to the fact that there's an enzyme that catalyzes that reaction, and they are actually activators, which is 2 ATP and N-acetyl glutamate. All right, the second step is the formation of a compound that is known as citrulline. So this takes place in the mitochondria, and of course it is released to the cytosol. Okay, so maybe that's why it is appearing like that. So carbamyl phosphate is reacted with a compound called ornithine. Okay, so ornithine comes from the cytosol into the mitochondria, combines with carbamyl phosphate, which we said was formed in the mitochondria, right? So again, I say that ornithine comes into the mitochondria from the cytosol and is combined with carbamyl phosphate to form this compound called citrulline. The enzyme there is called OTC, ornithine transcarbamylase. When I was little, many years ago, there used to be a bus service company that was called OTC. The reason why somewhere in Nairobi, there's still a place called OTC. It used to be the stage for those buses, and you know, they would go trans-country to all up-country destinations. So if you wanted to go to Nairobi, it doesn't matter where you live in Kenya, the most likely way you are thinking of getting to Nairobi is board OTC, then you get there. But the OTC we are talking about today is ornithine transcarbamylase, which is the enzyme that combines carbamyl phosphate and ornithine to form citrulline. Okay, so citrulline is then transported to the cytosol. Like I said, the reaction is taking place in the mitochondria. Okay, that combination of carbamyl phosphate and ornithine is taking place inside the mitochondria. Then, of course, we cross the border and we go to the cytosol, where the rest of the reactions will take place. So, ornithine, okay, which we said, of course, comes in from the cytosol to the mitochondria, just acts as a carrier molecule. It's like it comes into the mitochondria to carry carbamyl phosphate out of the mitochondria to the cytosol. As we shall see, this is a cycle. Okay, cycle will always begin at one point and will end at that point. If I tell you to run a complete path, a complete lap, maybe around the field, you have only completed the lap if you have gotten back to where you began. Okay, so for the urea cycle, because it is a cycle, it begins at that point where citrulline is formed. Okay, so ornithine will always be regenerated. I know, of course, you have learned about the TCA cycle, and you are always coming back to oxaloacetate. Then oxaloacetate combines with acetyl-CoA to form citric acid. Then you go around, then you come back again to oxaloacetate. So for the citric acid, where you always return to is to ornithine, okay? And then ornithine gets to the mitochondria, picks carbamyl phosphate, goes out back to the cytosol, runs the full cycle, you regenerate again ornithine, it enters the mitochondria. We shall see that cycle, okay? But, you know, form that picture in your mind that this ornithine that keeps coming to the mitochondria is to just serve as a carrier molecule. It comes to transport it comes to transport carbamyl phosphate, of course, out of the mitochondria into the cytosol. So, yeah. So, okay, synthesis of adenosine is the third step. So, citrulline, which we have seen, of course, how it is formed, is reacted with an amino acid called aspartate. Okay, aspartate is also known as aspartic acid. All right. So, the location now is in the cytosol. Okay, so we react citrulline and aspartate and we form adenosine. The enzyme in this case is called adenosine synthase, ASS. Okay, adenosine synthase. So for this reaction, we normally require 1 ATP. That ATP is normally converted to AMP and PPI, okay? Pyrophosphate. The way you read this in biochemistry, you say this is pyrophosphate, okay? So you know the relationship between AMP and ATP, you normally go from adenosine triphosphate to adenosine diphosphate to adenosine monophosphate. So this is adenosine monophosphate. What it means is, of course, the two phosphate bonds are broken. Okay. When you break one phosphate bond on ATP, you form ADP. When you break two bonds on ATP, you form. In adding compounds, we started with carbamyl phosphate, and we know how we generated the carbamyl phosphate. Carbon dioxide plus ammonia plus 2 ATP. Okay, that's how we formed carbamyl phosphate. So that one was already looking big. Then we added ornithine to form citrulline. Now we are taking that citrulline, we add aspartate, we form adenosine. So that must be looking quite big at that point. So the purpose of this aspartate is really to donate the second nitrogen atom. Go back to the ammonia that, to the urea that you drew. You know, at a certain point, I said you should take your pen, say NH2, C double bond, or oxygen bond NH2. Okay, so if you go there, we said we have two amino groups. So far, we have seen, you know, when we said ammonia, okay, ammonia is NH3, okay, NH4, sorry. So NH3, ammonium ion is NH4, okay, NH3. When we said ammonia plus carbon dioxide plus ATP, okay, so that's one of the nitrogen groups has come from there. Where does the second one come from? The second one is donated by aspartate. So at this point where citrulline is reacted with aspartate, the objective is to bring in the second nitrogen in urea. Okay, so aspartate actually is normally generated by transamination. This reaction here, glutamate plus oxaloacetate, forms alpha-ketoglutarate plus aspartate. is a reaction that you might have seen actually here, okay? This one here. In reverse, oxaloacetate plus glutamate, okay? Oxaloacetate is a keto acid, glutamate is an amino acid. The amino group of glutamate can be transferred to oxaloacetate. Oxaloacetate will become converted to an amino acid called aspartate, whereas glutamate will be converted to a keto acid called alpha ketoglutarate. Okay, so how aspartate is normally formed is, of course, through this transamination reaction, where we actually react glutamate with oxaloacetic acid or oxaloacetate, and we form aspartate. That's what we are saying at that point there, that glutamate plus oxaloacetate will give you alpha ketoglutarate plus aspartate. So this aspartate is really coming from oxaloacetate. Okay, yeah. And if you are keen, at the back of your mind, you are saying, I'm not seeing oxaloacetate for the first time also. I see it, I saw it somewhere in the citric acid cycle. Okay, that was always the beginning of the citric acid cycle, was oxaloacetate, where we combine it with acetyl-CoA. So the fourth reaction is argininosuccinate, which we said is a big compound, is now broken down, is fragmented into two components. The first component is called arginine, and the second one is called fumarate. Okay, fumarate is also not new. In the TCA cycle, we have also seen fumarate. You know, we went from citric acid, okay, to isocitrate, to alpha ketoglutarate, okay, then succinyl-CoA, then succinate, then fumarate, then malate, then oxaloacetate. Then, of course, now we went back again to that cycle, if you remember the TCA cycle, okay? So, again, fumarate is not a new compound. It is part of the TCA cycle, okay? So, and that is a very interesting observation that we are saying. In fact, the person who discovered the TCA cycle is called Krebs, okay? That's why we also call it the Krebs cycle. It is also called the tricarboxylic acid. It has three names: the TCA cycle, the Krebs cycle, the citric acid cycle, okay? Was discovered by Mr. Krebs. That Krebs is the same person who discovered the urea cycle. So he discovered these two cycles, two of them, because they have a connection, and the connection is actually through fumarate, okay? So, because as we have seen, fumarate is actually within the two cycles, connects the two cycles. So Krebs referred to the two cycles as the Krebs bicycle, okay? With each cycle forming, you know, one of the wheels of a bicycle. So, yeah. So the enzyme here, argininosuccinate, is hydrolyzed or is lysed by argininosuccinate lyase to form arginine and fumarate. If you wanted to speak like a biochemist, you say that argininosuccinate is hydrolyzed by argininosuccinate lyase to form arginine and fumarate. So that is step number 4. Step number 5, which is final, is now the formation of urea. Now, I should have said at that point, of course, that so this continues within the urea cycle, whereas that one joins the TCA cycle. Okay, so now we join the other wheel of the bicycle at this point. Okay, once we fragment argininosuccinate and we form arginine, this progresses within the urea cycle, whereas that one joins the TCA cycle. Okay, and you know, within the TCA cycle, what happens to fumarate? It is converted to malate, then oxaloacetate. Then we begin the TCA cycle again. All right, so here, the arginine, which we said continues within the urea cycle, is then now again hydrolyzed by another enzyme called arginase. So, of course, you will remember arginase. I don't know if there's arginase in this class. That's a different name, but this is arginase, okay? So arginase will break down arginine to form urea and ornithine. That's where we started, okay? So now we have completed the cycle when we form urea and we release ornithine. Remember, we are now releasing ornithine in the cytosol. What will happen to ornithine? It will diffuse into the mitochondria, react with a new carbamyl phosphate, form citrulline. Citrulline will diffuse out of the mitochondria back to the cytosol, react with aspartate to form adeninosuccinate, which we said was so big, then it fragments, we form arginine and fumarate. Arginine is then hydrolyzed, of course, by arginase, we get urea and ornithine. And the cycle continues that way, okay? So ornithine will return to the mitochondria to start the cycle again, as we have said. Now there is a cycle, all right? So we started somewhere. This compartment here that appears pinkish, that is the mitochondria, okay? So within the mitochondria, we said, we start by reacting carbon dioxide and ammonia. This is ammonium ions, okay? So in the presence of an enzyme called carbamyl phosphate, the CPS1, then we form carbamyl phosphate. Of course, you remember what else is required there, okay? Yeah, so then you form carbamyl phosphate. Carbamyl phosphate is then reacted with ornithine, which we said comes from the cytosol, comes into the mitochondria. It is combined with carbamyl phosphate by an enzyme called ornithine transcarbamylase. We form citrulline, which then diffuses out of the mitochondria into the cytosol, okay? Citrulline is reacted with aspartate. The enzyme there is adeninosuccinate synthase. We get the B complex, adeninosuccinate, fragments by adeninosuccinase. Fumarate goes to the TCA cycle. Arginine continues the urea cycle. It is hydrolyzed using arginase. We form urea and ornithine. The cycle begins again. Okay, so that is very interesting. I hope no one is finding that one difficult. I hope you are finding it interesting. And by the way, that is what is happening in your liver cells as you listen to me right now. Okay, your liver cells have no problem doing this over and over and over since you were born. Okay, so it cannot be difficult for you to know it if in fact you are doing it over and over and over again. So the urea cycle, which is what we have just seen, but now in structures, okay? I will not be examining you for the structures here, but it is good for you to look at them. You appreciate this, okay? If you are biochemistry students, okay, that is what you would have to do. Learn to draw those structures, look at the changes as they occur. But even now, you can actually look at them and see, appreciate that. So there is 2 ATPs, then there is bicarbonate. This is the bicarbonate that Dr. Mayo was talking about, bicarbonate. So carbon dioxide actually is used to form this. When you combine carbon dioxide and water, you form weak carbonic acid, which then dissociates to form bicarbonate and hydrogen ions. Okay? Somebody want to say something? Jemima. Mwikali, are you saying something or just, yeah. Okay, so 2 ATP plus bicarbonates plus ammonia in the presence of, what did we say the name of the other activator was here? What was it? We have bicarbonates, ions being combined with ammonia in the presence of one of the activators is ATP. What is the other one?
Glutamate.
Huh? Luis?
Glutamate.
I didn't hear you well. Say it again.
Acetyl glutamate.
Acetyl glutamate. Good. Okay, so now, so then we have the formation of carbamyl phosphate. So in case you are just thinking in your mind, how exactly that does look like? So you can see the ammonia here, then carbon, double bond oxygen. So now that you know the structure of urea, which is actually here, okay, you know that we have started forming the urea. All we need later on is to attach an amino group there, okay? So we've already formed part of the urea, and you can see that C double bond oxygen is coming from bicarb, whereas of course NH2 is coming from the ammonia, okay? So we form carbamyl phosphate, Then, of course, we reacted it ornithine that came from the cytosol. Citrulline would look like this. Okay, so we say this citrulline is really ornithine. Sorry, is really a carrier molecule because this doesn't go. It is not incorporated into the urea structure. Okay, this part here is what is incorporated into the urea structure. So that comes then out of the mitochondria into the cytosol, where it is reacted with aspartate. So you can see aspartate is in red here. So this ammonia group here, all right, if you were to track it slowly, is what will become this ammonia that will be added there. Okay, so when aspartate combines with citrulline, we form the big compound which we said is called argininosuccinate. Okay, on this argininosuccinate, if you are keen, you will see that okay, there is this NH2. Although now we have a double bond carbon, okay, and then we have NH here, okay. So you are following it that way. Then we shall remove fumarate, okay. Fumarate really will mean that we have cut here, so that we retain that up there, okay, which is right here. And you see that? So we cut here, that is released as fumarate. That fumarate we said will go to the TCA cycle. Now we are left with a compound that is called arginine, okay? So this arginine here will be hydrolyzed, okay? To hydrolyze means to cut a bond using water. You can see the water coming in here. So we will hydrolyze this bond here. This oxygen here is what we will add here. Double bond, carbon double bond oxygen. Where the oxygen will come from is from this water molecule here, okay? So we'll hydrolyze that. And now we shall have formed urea, and the rest of it is the ornithine again, which goes back again, to the mitochondria and repeats the cycle. Okay, so you can take your time later on, of course, just look at it to appreciate it. If you want to look at the structures and try drawing them for fun, that is good. But what you must make sure you know, of course, is that you actually know the names of these compounds. We call them metabolites that are participating. You know very well that you can say that we started with ATP, bicarb, and ammonia, and we formed carbamide phosphate. Then we went to citrulline, argininosuccinate, fumarate, like that. So what is reacted with what, and which enzyme is involved? Those ones you actually have to know them. Okay, so back here, we had all the names of the enzymes. This one here, this one here, this one here, that one, and that one. Those are the enzymes, of course, of the urea cycle. So you need to know them and the steps that they actually catalyze. Okay, so recycling of fumarate in the citric acid cycle. So it's important, of course, to just look at that. So here we said citrulline. Remember, this is the urea cycle here, or I should point it in that direction. Okay, where of course we saw ornithine always coming in to combine with carbamide phosphate within the mitochondria to form citrulline, which is then released. Citrulline is then reacted, of course, with aspartate to form argininosuccinate. Then we said now argininosuccinase will actually hydrolyze this, and we release fumarate and arginine, which continues in the cycle. That's what we said. Whereas fumarate joins the TCA cycle, the citric acid cycle, okay? So fumarate is released, it is converted to malate, then malate to oxaloacetate. Then what happens? Remember that reaction that we were talking about. Oxaloacetate is transaminated back to aspartate. At a certain point, we said where aspartate comes from, okay? It comes from the transamination of oxaloacetate, where oxaloacetate is reacted with glutamate. We form aspartate and alpha-ketoglutarate, okay? But it's interesting if you look a little bit further, because where is this glutamate actually coming from, okay? Because glutamate is the one that converts this oxaloacetate into aspartate. Then we go back again to the, aspartate once it is formed, comes back to the urea cycle, it reacts with citrulline to form argininosuccinate. It fragments, we release fumarate. We go around this cycle, we regenerate aspartate there, okay? goes back again like that. So there's a very interesting cycle here. What is it actually doing is the question. If you look carefully, you will see that somewhere here, there are actually amino acids that are constantly being broken, okay? And amino acids are normally broken down through transamination reactions, as we have said. So when you remove the amino group of these amino acids, you form keto acids, okay? That amino acid that you are removing, you give it to alpha ketoglutarate, okay? That's a transamination reaction. You give it to alpha ketoglutarate, which becomes converted to glutamate. Then glutamate now gives that amino group, again, it came from amino acids, gives that amino group to oxaloacetate, it becomes converted to aspartate, okay? Now aspartate joins the urea cycle. So essentially what we are saying is... Jemima Mwikali and your friend. Can you mute yourselves? Jemima. Yeah, the exam will come. I hope you will not be writing what you are saying. Okay, so what I was saying is, when you look carefully, therefore, you realize that this aspartate that we said brings in the second amino group is actually also getting that amino group from breakdown of amino acids. Just like, of course, the ammonia that we were adding in the first reaction here, okay, to form carbamyl phosphate. Remember, we said ammonia is combined with bicarbonate in presence of ATP. Where is that ammonia coming from? If you look at it carefully again, it is coming from the breakdown of amino acids. As amino acids, you remove the amino group, you form a keto acid. That amino group is given to alpha ketoglutarate, becomes glutamate. Now we release that ammonia from glutamate. We say we form free ammonia, of course, in the liver, okay? Glutamate dehydrogenase, we say it does that. Now, that begins the urea cycle. On this other side, other amino acids, of course, are being broken down, and the amino groups being transferred, of course, to glutamate, finally to oxaloacetate to form aspartate. All right. Then that aspartate brings those amino groups into the urea cycle. Okay. So this pathway here serves the same role, of course, as what we are seeing here, just bringing in amino groups, because ultimately urea will have two amino groups, NH2, CO, and then another NH2. Okay, so you will take time later on, of course, to just be able to look at that and understand it well. Yeah, so the overall equation, therefore, for the urea cycle is carbon dioxide plus ammonia plus 3 ATP plus water gives you urea plus 2 ATP plus pyrophosphate plus plus 2 inorganic phosphate plus MP plus pyrophosphate plus fumarate. Okay, that's the overall equation of the urea cycle. So urea synthesis is an energy demanding process. Why do we say that? We have seen, of course, that you need quite a number of ATPs, okay, 3 ATPs. So the equivalent of 4 ATPs are consumed for each molecule of urea that is synthesized. Okay, so CPS, now we are talking about the regulatory aspects. Carbamyl phosphate synthase. CPS is carbamyl phosphate synthase. So CPS is normally allosterically regulated. Allosteric regulation means, of course, you know, there is a way you bind certain compounds onto the enzyme to activate it or to actually inhibit it. Okay, so we said acetyl-CoA, maybe we haven't said that. We talked about acetyl glutamate as being an activator. Okay, so N-acetyl glutamate is the compound that binds to CPS. Okay, and activates it. You remember we said earlier on that we need ATP and then we also need N-acetyl glutamate. So that N-acetyl glutamate is normally formed from acetyl-CoA and glutamate using an enzyme called N-acetyl glutamate synthase. So this one here, of course, I haven't labeled it, but this is the N-acetyl glutamate that we are talking about. That is an activator, of course, of the formation of carbamyl phosphate. For us to go from ATP plus ammonia, we need, bicarbonate plus ammonia plus ATP, we said we needed N-acetyl glutamate, of course, as an activator. And also the ATPs are actually activators. Yeah, so so this is the reaction we are talking about to form carbamyl phosphate. We require the activity of N-acetyl glutamate. All right. But the other interesting thing is that reaction that we talk between acetyl-CoA and glutamate to form N-acetyl glutamate is normally activated by arginine, which an arginine itself is a compound that is formed in the urea cycle. Okay. So when the urea cycle begins to run and we form arginine, that arginine becomes useful in activating the formation of this compound, N-acetyl glutamate, from acetyl-CoA and glutamate. Okay, from acetyl-CoA and glutamate, we form N-acetyl glutamate. Okay, so then that N-acetyl glutamate activates, okay, the urea cycle by beginning it, okay, because this is the beginning of the urea cycle, converting this one into carbamyl phosphate. Then carbamyl phosphate runs again in the cycle. Part of what is formed there is arginine. So this is really positive feedback. As we form more and more intermediates of the urea cycle, the more activated the pathway becomes, because we will always be forming more and more arginine. That arginine will be activating the formation of N-acetyl glutamate, which will be activating the initiation of the cycle. Okay, so we refer to that as positive feedback. Okay, now we talk about the disorders of the urea cycle. And one of the things that I would like you to know is within the functioning of living systems, reactions are catalyzed by enzymes, and these enzymes are very useful, very important actually. Because otherwise, those reactions will not take place at a rate that can sustain life. There are so many reactions that take place in each of your cells. In the absence of enzymes, those reactions cannot take place at a rate that will sustain your livelihood. You will actually die if something happens to those enzymes. But of course, sometimes, it's not all the enzymes that are affected. Maybe one enzyme is mutated, the others are okay. So everything else is running well except at that particular point. So that one is a disorder. Okay. So like now the urea cycle, we saw of course that we had 5 enzymes. The 5 enzymes are listed here, from carbamyl phosphate synthase, ornithine transcarbamylase, which I said was OTC, argininosuccinic acid synthetase, and then argininosuccinic acid lyase, and then arginase. Okay, so these enzymes need to be present in appreciable amounts for the urea cycle to be able to run. But if one of them is deficient, then we end up with a disorder. So we have disorders that are listed here. So we are saying, for example, if there is a deficiency of carbamyl phosphate synthase, we end up with a disorder called hyperammonemia, and also hyperinsulinism. Okay, so the chromosome here that is listed is where, of course, this particular enzyme is coded for within the human chromosomes. Okay, yeah. The same applies to ornithine transcarbamylase. If it is deficient, those are the 2 disorders that we end up with. If it is this one, we end up with those 2 disorders, like that. Yeah, so most important is you focus here, the enzyme and the disorder. These ones are okay, you can look at them, but it's the disorders here that you need to focus. The deficiency of arginase would lead to congenital arginemia. Okay, so that's it for urea, but maybe we need to remind ourselves that then ultimately, of course, that urea, once it has been formed, is going to be filtered. Again, that reaction was taking place in the liver. Once urea has been formed in the liver, It diffuses into blood, then blood goes all the way to the kidneys. It is filtered. That urea is put into urine, then out of the system. So some of the nitrogen compounds that are in urine, one of them now, of course, obviously is urea. But we also normally have a certain amount of ammonia produced in the kidneys by the amination of glutamine. Okay, so reduces the body's acidity because the process removes protons. Okay, so when we say the process removes protons, of course, you know ammonia, NH3. For you to go to NH4, you actually add hydrogen ions. Okay, hydrogen ions are what we are referring to as protons. So NH3 plus protons or plus hydrogen ions forms ammonia in the kidneys, and then we can release that ammonia also in urine. So you don't only release urea in urine, you also release ammonia. And then, okay, we have seen the role. Makes urine, makes the body less acidic. Okay. One of the challenges that our bodies face every day is to maintain the correct pH. You don't want it to be too acidic. You don't want it to be too alkaline. Okay. If you accumulate a lot of hydrogen ions, the body becomes acidic. So if you can remove as much of that, then that is good. Then we have another one called uric acid, which is really not key for today's discussion, but it is important for you to know that uric acid, which is the final metabolic byproduct in the breakdown of purine nucleotides, is also normally released, of course, in urine. This uric acid, if it is not removed from the body, it normally accumulates, especially in the joints, and causes a lot of pains. Okay, not so much among young people like yourself. Okay, but when you become middle-aged and old-aged, you have seen, of course, people, they say, I have arthritis, you know, those kind of things. So basically, what is happening is you have inflammation in their joints. This uric acid accumulates, it crystallizes, forms salts actually. So you can imagine, you know, salt inakaanga kama machupa chupa yenye pasuka, sindio? The salts, the crystals. So you can imagine having those ones within the joints. The joints normally have a fluid called synovial fluid, which is supposed to make the joints just move smoothly, easily, you know. You can dance without a problem. You can do squats and what without a problem. Because there is lubrication in your joint by the synovial fluid. But if you begin to have accumulation of crystals, in this case, we are talking about uric acid crystals, it becomes very painful, okay? So that's how gout actually normally forms. Yeah, so uric acid also is a nitrogen compound. You can see it has nitrogen there, it has nitrogen, it has nitrogen, it has nitrogen. Just like we saw urea has nitrogen, ammonia also has nitrogen. The other compound that has nitrogen and is a waste product that is removed in urine is creatinine. So creatinine comes from skeletal muscles, okay? It plays a very important role, not creatinine itself, but creatine, as we shall see. Creatine plays a very important role in muscle metabolism, especially in powering the muscles, okay? So usually the body system is powered by ATP, as you know. The energy currency of your body system is ATP. But there's another compound that also powers the muscles that is called creatine phosphate, okay? So creatine phosphate, we said, when it ages, when creatine phosphate ages, it actually becomes creatinine. So creatine phosphate ages either through dephosphorylation or, of course, dehydration, okay? You form an ethanol anhydride here that is called creatinine. Creatinine is no longer useful when it is formed. It is a byproduct that needs to be eliminated. But importantly, you notice that it contains nitrogen, okay? So it is a nitrogen-containing metabolic waste product that is then put into urine and removed from the body. So now let's talk about that creatinine, okay? But before we talk about creatinine, we have to say something about creatine because we have said that is where it begins. So creatine plays an important role in the muscles. It actually does what we call energy buffering in the skeletal muscles. So creatine is normally synthesized in the liver, the kidneys, and the pancreas. After it has been synthesized, it is transported to the muscles where it is stored in the form of creatine phosphate. Okay, so creatine is converted to creatine phosphate. That's what we are saying. How do you convert creatine to creatine phosphate? You react creatine and ATP. Okay, creatine plus ATP gives you creatine phosphate. Okay, that creatine phosphate is a high energy molecule. Okay, what do I mean? I mean when you break that bond between creatine and phosphate. Okay, because creatine phosphate is just creatine bonded to phosphate. When you break that bond between creatine and phosphate, you release a lot of energy, which can power the skeletal muscles. So this creatine phosphate acts as a rapid energy reserve during short bursts of activity. So when you need a very rapid supply of energy for your muscles, you can imagine where you are seated. I don't know where you are. If something came in there, let's say you just stand and you saw that, oh, somehow there is a cobra that is just about to pounce on you, okay? The way you will find yourself outside that room, you may not be able to explain for the rest of your life, okay? You will just know that you somehow got out of that, even if the door was locked, even if the window is too tiny, somehow you find yourself outside. The muscles will be given a very rapid burst of energy, okay, that will be able to drive your activity, to be able to move out of that place so fast, okay? So usually between 0 and 4 seconds, that rapid burst of energy that you need between 0 and 4 seconds is given to your muscles by of creatine phosphate. Okay, so creatine phosphate, therefore, is a very important compound for muscle activity. So, however, of course, so it supports muscle contraction, especially in high intensity efforts. So excess creatine is then converted to creatinine, okay, which is actually a waste product. Okay, so before we go again to that conversion, let's say, how is creatine actually? We have seen it is synthesized in various tissues, okay? We have indicated here the liver, the kidneys, and the pancreas. That is where creatine is synthesized. But what is it synthesized from, okay? So creatine is actually synthesized from amino acids, arginine, glycine, and methionine. Oh, okay, somebody has just remembered that we were talking about arginine in the urea cycle. Kumbe, it is actually an amino acid, okay? So some of these amino acids, you know, they play multiple roles. It is not just a single role. Aspartate, we also talked about it. We know of aspartate being incorporated into proteins. When we are making proteins, we use amino acids, okay? But so amino acids don't just play that role of being incorporated into proteins. There are other functions that they play, including forming neurotransmitters, okay? So neurotransmitters, as you know, are very important for communication between different components in your body. Okay, you have the nerves, and then you have a nerve junction between two nerves. The two nerves, they don't touch each other. There's a synapse. How is information relayed across the synapse? You have compounds that are called neurotransmitters. Most of them are actually formed from amino acids. For example, tryptophan is used to form serotonin. Tyrosine is used to form epinephrine and norepinephrine. If you like, those are called adrenaline and noradrenaline, okay? And another one called dopamine, okay? So amino acids are amazing compounds. The functions they play within your body is enormous, okay? So arginine here can be used actually to also make creatinine. You combine arginine and glycine and methionine, although that methionine actually has to be converted to a form that's called S-adenosine methionine. Okay, so the first step in the formation of creatine, and you have to pay attention because it's only 2 steps. So by the time you are saying, okay, let me now listen, we are finished what we were actually saying, okay? So arginine plus glycine are reacted to form guanidino acetate. So the enzyme there is called glycine amidotransferase, AGAT, okay? Arginine glycine amidotransferase. Arginine glycine amidotransferase, which is called AGAT. So this actually occurs in the kidneys and in the pancreas. So once we have formed guanidino acetate, we move to the second step, where now that guanidino acetate is reacted with some And I said SAM actually comes from methionine. So methionine is first converted at a certain reaction there to form SAM, and SAM in full is S-adenosyl methionine, S-adenosyl methionine. And the function of S-adenosyl methionine, usually in biochemical reaction, is to donate a methyl group, CH3. CH3 is a methyl group. That's all we need actually from this SAM here to give us CH3, which is a methyl group, which we then add to guanidino acetate and we form creatine. So creatine is formed in just 2 quick steps. Step number 1, we form guanidino acetate. Then we methylate guanidino acetate and we form creatine. Okay, so the enzyme there is called guanidino acetate methyl transferase. You understand that because actually transfers a methyl group from SAM onto guanidino acetate. So this occurs in the liver. Okay, the first 2 reactions we saw occur in the kidneys and the pancreas. Now this final reaction occurs in the liver. The reason, of course, why we said inter-organ collaboration in energy metabolism. Actually, even in synthesizing this creatine, we work between organs. Okay, we form a certain intermediate in the kidneys and in the pancreas, which we have said acetate. Then it goes all the way to the liver where we methylate it, and now we have creatine. Okay, so creatine is then transported to the muscles. Okay, so you see organs are working together here for storage as creatine phosphate. So that creatine has to be phosphorylated using ATP. The enzyme that phosphorylates creatine to creatine phosphate is called creatine kinase. I may not have mentioned it in these notes here. Oh, it is there. Okay. So creatine kinase converts creatine to creatine phosphate, of course, using ATP. So summary of what we have said for the synthesis of creatine. Converts, of course, that to guanidino acetate. Now, see, this here is an interesting compound. This one we know of. It's called ornithine. Okay, so ornithine, we encountered ornithine somewhere. Okay, where did we encounter ornithine? Someone? And what was it doing? Sema bwana, tuendelee.
The urea cycle.
Yes, Vanessa says in the urea cycle. Yeah, so we encountered that in the urea cycle. And it is interesting now we are talking about the formation of creatine and then we encounter ornithine again. So this ornithine is exactly the same compound. It actually, from here, it joins the urea cycle. Okay, so the reason why we were talking about the 2 actually today, why did we talk about urea cycle and then we talked about creatinine formation? There's actually this link through ornithine. Although we have also seen arginine as a connection. So, so we form guanidino acetate. Then guanidino acetate is methylated using S-adenosyl methionine, which we said comes from methionine. So methionine is first converted to S-adenosyl methionine. Then S-adenosyl methionine donates a methyl group to guanidino acetate using an enzyme called guanidino acetate methyl transferase. And by so doing, we create creatine. So there's 2 steps. First, formation of guanidino acetate. Then from guanidino acetate, we directly form creatine. Yeah, so once we have formed the creatine, we said it will have to be transported. That was happening in the liver. Once we have it is transported to the muscles. In the muscles, creatine is phosphorylated by creatine kinase, okay, which requires ATP to form creatine phosphate. Now, this creatine phosphate is what we said is a very powerful, we say it's a phosphagen, okay? It's a very powerful energy currency which can power your muscles, you know, for the rapid energy, intense energy bursts that you actually require, all right? So, however, that creatine phosphate and even creatine itself normally undergo aging, okay? So, creatine can undergo dehydration to become creatinine, whereas creatine phosphate is dephosphorylated and it becomes creatinine. So creatinine is being formed constantly from, excess creatine and aged creatine phosphate. So creatinine is then filtered into blood. From blood, of course, it is transported to the kidneys, the glomerulus that is actually in the kidneys. It's a part of the nephron, the kidneys, which then filters that creatinine, puts it into urine, and then, of course, now that can be gotten rid of from the body. Okay, so the story of creatine and creatinine is very interesting, but short also, so it's very difficult to forget. The synthesis pathway, once you form the creatine, how you activate it is just in one step. You have creatine phosphate. Then how you inactivate. When we talk about inactivation, we mean degradation. Okay, how this creatine phosphate is actually degraded to form creatinine is usually just a single step: removal of phosphate, removal of water. Okay, so what you form, this creatinine, creatinine is actually referred to as an internal anhydride. Let me see. There are people who are asking me questions, or were they responding? Yes, yeah, somebody is asking, creatine phosphate is stored in the muscles. Yeah, that's what we said. It is stored in the muscle. Of course, storage here means, of course, until you need to use it. Okay, so it is in the muscles until you're actually required to use it. But when you use creatine phosphate, you break it down to creatine and phosphate. Then that creatine is rephosphorylated again using creatine kinase. You form creatine phosphate, which you will break down again. Of course, that cycle is repeated over and over. Where is methionine used in the synthesis? Where is methionine used in the synthesis? The answer is right there. Methionine is first converted to S-adenosine methionine. Okay, and then S-adenosine methionine donates a methyl group to guanidino acetate, and by so doing, we form creatine. Okay. So this, of course, slide just summarizes that conversion now to creatinine. So creatinine is formed from creatine phosphate in muscles. The reaction is usually spontaneous and non-enzymatic. Okay, so you don't need an enzyme to convert creatine to creatinine or phosphocreatine to creatinine. It just occurs spontaneously, non-enzymatically. But it happens at a very constant rate. So the rate of creatinine that you form within your muscles is proportional to your muscle mass. So the more muscular you are, the more creatinine you form. But that also tells you that men will therefore tend to have higher levels of creatinine in blood and in urine compared to women. And the reason is because men would have more muscle mass. It will also tell you that children will have less compared to adults, again, because of less muscle mass. Okay, so, but otherwise, for a given individual, we are saying the rate of formation of creatinine is actually constant. So that creatinine is not reused or metabolized within your body. It then becomes a waste product, which is transported via blood to the kidneys, and then it is excreted. It serves as a biochemical marker for muscle metabolism. So sometimes, of course, people can have issues with their muscles. Muscles are aching or undergoing some kind of inflammation, degradation. One of the markers that can be requested for and monitored in the labs is usually creatinine. So creatinine can be measured in blood. It can also be measured in urine. But the other application, of course, of not just creatinine, but also urea is in the assessment of the functioning of the kidneys. That is referred to as renal function tests. So if someone goes to the hospital and the symptoms which they present with, the doctor thinks they could be having kidney problems, okay? One of the things that they will request to be analyzed in their samples, and the sample can be blood or urine, is the levels of urea and the levels of creatinine. So urea and creatinine are routinely measured as part of renal function tests, okay? They indicate whether the kidney is doing its filtration work appropriately, okay? So both are waste products excreted by the kidneys. And again, when you think about it, why would we be using them? Because the kidneys is the one that filters them and removes them from the body. So if they are accumulating in the body, it is because maybe that kidney is not working well. Otherwise, if it is working well, they are not supposed to accumulate. The levels should remain within a certain range, which of course you can refer to as a normal reference range. Yes, Prof. Can I come in a minute? Okay, so they would actually normally be maintained. I mean, urea and creatinine are normally maintained within your blood at a certain range that is referred to as the normal reference range. When they go beyond that, it means, of course, the kidneys is not filtering them well. So now you begin to say, maybe this person has a kidney problem, okay? So as part of renal function tests, you measure the levels of urea and creatinine. There are other things that you also measure, which we are not going to talk about. They include electrolytes, sodium, potassium, among others. So urea levels depend on protein intake, liver function, and renal excretion. So if you are looking at the results, you must always also remember that, oh, by the way, it's not just the kidneys that matters here. The liver also matters. Why? Because urea is synthesized in the liver. The diet of that person also matters, because if they are eating a lot of proteins, when they are broken down to amino acids, they are deaminated, they form ammonia, which is then converted to urea. So they could be having a lot of urea because of a high protein diet. And then, of course, finally, if they are having issues with the kidneys. Elevated levels may suggest reduced kidney filtration, but you have to look at it together with the symptoms that they're presenting with. So we therefore use urea and creatinine to monitor what we refer to as glomerular filtration rate and monitor renal health. Okay, so thank you. That is all I intended to talk about, unless someone has